Today let's look at the plant cell. The cell is the smallest unit of life, and cells come from pre-existing cells. Those are the two criteria of cell theory. What's amazing is that almost all of the plants that we've been studying this semester and will continue to study are multicellular organisms. So we really do want to get down to that smallest unit of life, the cell, and understand how all of these plants, these multicellular organisms that we've been studying, what they're like at that smallest unit and what those structures are. So our plant cell is going to be very geometric in shape, and the reason for that is because the plant cell has that sturdy cell wall. So I'm going to start by drawing the cell wall here in black. You can see it has a very geometric shape, and hopefully you've been recognizing this as we've seen things underneath the microscope so far this semester. When a plant first starts growing, and if you think back to your fast plants, when those cotyledons first came out of the seed coat and started to spread out and show themselves to the world and start doing photosynthesis, those cells were dividing inside of the cotyledon, and the way that they were dividing is they were doing mitosis, where one cell makes two genetically identical cells, and those new cells had a very thin cell wall. That primary cell wall doesn't have a whole lot of cellulose, doesn't have a lot of those tough lignin fibrils. So that basket that surrounds the water balloon of a cell early on in that cell's life tends to be really, really, really fragile and really delicate. This is part of the reason why we love to eat baby lettuces, or to eat sunflower shoots, or the radish seedlings, or all of the things that we call sprouts. There's a lot of different species that are considered sprouts, and we put them on sandwiches or salads. We love those really young cells, essentially. They're easier to eat and they're easier to digest. Now, as the cell matures, the plant's going to invest more energy in making those cellulose fibrils, in making lignin. And that's what makes the cells so strong. That's what makes them tough. And at least our mouths can make things very chewy, very just rough to eat through when we're eating some of the more mature plants. Now, if you want to think about the cell walls in a tree, now there's some secondary cell walls that are extremely, extremely tough. So think about oak trees, can last 300 years. Redwood trees, 1,000 years, 2,000 years, almost 3,000 years. And that cell wall can stay sturdy, can stay strong. So keep that in mind as we go through this unit. We're going to keep touching base on those cellulose fibrils and the role of the cell wall. But for today, we're just going to draw our cell wall, and we'll assume that it's a secondary cell wall. Now, in order for cells to stick together, there's going to be this really nice sticky substance in between our two separate plant cells. Here in between is going to be our middle lamella, and that middle lamella is made of pectin. Now, I love to make jams and jellies, and I love to manipulate that pectin in between the cell walls. So essentially what we do when we're making jams and jellies is we try to get rid of as much of the cell wall as possible. We want to have just that beautiful juice, and really the juice is going to be sugar and water, and whatever flavors we associate with the plant, whether that's the strawberry flavors for strawberry jam or grapes for grape jam, whatever. So we get a hold of that sugary water, we add in some pectin, it thickens it up, and it makes it into those jams and jellies. So our middle lamella has the pectin. In the cell wall will be a bunch of holes, and those holes will be pits. And the pits are going to allow molecules to move in and out in between the cell wall, potentially interact with the plant cell. Well, I'll get to that here in just a minute. And then sometimes connecting our pits will be this kind of cytoplasmic connection called the plasmodesmata. And what this allows cells to do is talk to one another, at least molecularly. And one thing that will happen is an insect will start to eat a leaf. Say a caterpillar is chomping along. And the plant needs to be able to send the signal from one cell to the next. Essentially, one side of the plant has to say, hey, I'm getting eaten. You on the other side, you may want to be ready for this. Maybe you're getting eaten too, or maybe the caterpillar's going to walk over there. And so the plant can send very quickly signals from one cell to the next through those plasmodesmata. We've often had this point of view that plants aren't really doing a lot of interaction with the environment. They aren't really communicating within the body. The cells are not communicating, and the plants themselves in the larger environment are not communicating. And that's a paradigm that we've had to revisit and change, because it turns out that there is a tremendous amount of communication from cell to cell, and even from plant to plant in the community. The other thing that plants can do is they can send electrical signals. Some of them are going to send electrical signals from one cell to the next. And that's a whole other area of research that we're not really going to touch base with. I guess what I'm trying to drive home here is the idea that, yes, the cell is the smallest unit of life. When cells bond together to make tissues, tissues come together to make organs, whether that's a leaf, a stem, or a root, and eventually we'll get the entire plant body. But communication, interaction, is happening at the cellular level. It's happening all the way at the organ, all the way at the whole organism, all the way at the whole ecosystem. No organism on this planet exists without any interaction with the environment. Same holds true for plants. So we've got our cell wall, our middle lamella, our pits, the holes in the cell wall, and then our plasmodesmata allowing two cells to communicate with one another. The next structure that's going to be important in our plant cell is going to be that cell membrane. I'm drawing it a little bit exaggerated here, just so that you can get that feeling of where the cell membrane is, and also see the label, I hope, as well. The cell membrane is a plasma membrane. It is the defining boundary of the cell. Now, in plant cells, what's going to happen is this cell membrane is actually going to be pushed up tightly against the cell wall. So when you're looking at a plant cell underneath the microscope, it's impossible to tell the cell wall away from the cell membrane. And that's kind of intentional. The water balloon of the cell is going to be constantly pushing against the cell wall basket. It needs to stay expanded. It needs to be pushing against that cell wall. This helps plants to stand upright against gravity. So our plasma membrane, our cell membrane, gets pushed up tightly against the cell wall. Think about a water balloon inside of a basket, and that water balloon is completely inflated. Now, the cell membrane is going to be made up mostly of phospholipids with those hydrophilic water-loving heads and the hydrophobic tails that are repelling water. We'll have two of them together, and they will be making that bilayer. In our cell membrane, we're going to have all kinds of proteins, and those proteins are going to allow this cell to be able to let molecules in and out, to communicate with other cells. Those proteins are going to be hugely important. We'll have some cholesterol in there. Weird thing to think about, the idea that a lot of plant cells have cholesterol. Excuse me. And then we're going to have some additional carbohydrates here in our cell membrane. So we've got our cell membrane, our cell wall. Now let's talk about the parts on the inside. Way up over here, I'm going to draw a great big circle. This is essentially the nucleus. The nucleus is almost perfectly round and has a very smooth edge to it unless the cell is dividing. So our nucleus is just a plasma membrane with DNA inside. When we have loose DNA, looks like hair, we call that chromatin. If our cell is dividing, that DNA has been packed tightly into a chromosome. And generally when we draw a chromosome, it looks like a hot dog. So I'm going to erase the chromosome out of here so that we can just focus on our cell parts. So we've got our nucleus, our plasma sac of DNA. Connected to that is going to be the plasma membrane sac called the rough endoplasmic reticulum. Now, the rough endoplasmic reticulum has lots and lots of folds, and the reason why this essentially sac of biochemistry is so deeply folded is it gives us lots of surface area for the little ribosomes to be attached. The ribosome is going to read mRNA and make a protein. So inside of our rough endoplasmic reticulum, it's going to be a plasma membrane sac with a biochemistry for folding proteins. There's a second type of endoplasmic reticulum. It is attached to the rough endoplasmic reticulum, and it too has lots of folds, lots of surface area, but doesn't need all that surface area. At least not for the same reason. The smooth endoplasmic reticulum is going to be making lipids. It's also going to detoxify the cell. So another organelle that we will have inside of our plant cell will be the mitochondria. The mitochondria has two separate plasma membranes. Inside of the mitochondria, aerobic cellular respiration can happen. Essentially what's going to happen is broken glucose will get turned into ATP energy. Now, one of the things that I hate about learning about a cell is a textbook will say things like, the mitochondrion is a powerhouse of the cell. And that's just an awful analogy for helping us understand and remember what this organelle can do. When we call it the powerhouse, we first have to think of like Byron Nuclear Power Plant, making electricity. And that electricity is energy, and then that energy does everything in our house. So if we have an electric stove and an electric dryer or electric heat, that all of that energy is helping us to live inside of our little boxy home, our little cell. And it's awful, because first of all, most of us aren't familiar with or don't initially think about this flow of energy through our lived systems, through our built human systems. So avoid those types of analogies. You'll notice that I don't use them. That's intentional. If you are, if you're remembering the mitochondrion as doing aerobic cellular respiration, that's a much more accurate way to remember what this organelle is doing. So the reason why we need to know and be accurate with what the mitochondrion is doing is because of what plants can do. Plants are autotrophs. They're self-feeders. So not only do they do photosynthesis and make glucose out of sunlight energy, but then they can immediately use that glucose, run it through the mitochondrion organelle, and do aerobic cellular respiration and make lots and lots of ATP. So this would be the equivalent of you or I just sitting outside on a sunny day and being full. Like, we ate our dinner already. We sat outside for 20 minutes. We wouldn't have to do all of this, run to the grocery store, bring it home, cook it up. We could just lay there and make our own energy. It's essentially what plants are doing. It's absolutely brilliant and amazing. So clearly the most important organelle from our point of view is going to be that chloroplast organelle. Now, chloroplast organelle has three membranes. An outer membrane, an inner membrane, and this weird third thylakoid membrane. We're going to learn about that in more detail in Chapter 4. The chloroplast organelle is doing photosynthesis. It can capture and use that sunlight energy to make glucose. Now, at this point in the semester, I want you to get the formula for photosynthesis memorized. CO2 plus H2O gives us glucose. And yeah, we happen to make some oxygen. I do also want you to start memorizing that formula for aerobic cellular respiration. Glucose and oxygen make carbon dioxide, water, and ATP energy. If you start getting those formulas stuck in your head now, it'll make the Chapter 4 cellular metabolism a lot easier. The other important thing about the chloroplast organelle is the chloroplast organelle, being the most important organelle inside of a plant cell, this is going to be moved around by the motor protein. And what the motor protein is going to do, a motor protein is just a protein that walks around inside of the cell, and it walks on these microtubules that crisscross back and forth. I would love to draw them, but I think what's going to happen is this diagram is going to get real messy real quick. So I've got all these microtubules all over inside of the cell, and the motor protein is going to be dragging around any organelle. It could drag around a mitochondrion, it could drag around a vesicle, it could do all kinds of things. But in a plant cell, a motor protein that has a chloroplast organelle is going to be participating in cytoplasmic streaming. So on a really bright, sunshiny day, with all of that intense light hitting down on the plants, on the surface of the leaves, what often happens is that intensity of the light can get to be too much. Now, most of the time we think about it, like plants never get enough sun, right? They need lots and lots of sun. But the thing is that sunlight, when it's too intense, will damage the proteins inside of the chloroplast organelle and stop photosynthesis from happening. So anytime it's above 85 degrees Fahrenheit in the ambient air temperature, if it gets to be 85 degrees inside of the leaf, those proteins inside of the chloroplast organelle will change shape and they'll stop working altogether. They'll denature. And so what our little motor protein is going to have to do is it's going to have to hide this organelle, and it will often hide the chloroplast organelle underneath another organelle. So in our plant cell, most of the cell space is often going to be dedicated to a vacuole. And the vacuole is just this generic storage organ. It's a plasma membrane bound sac, and our plant really could put anything in there. Most of the time it's going to be putting water in there, which is why I drew it in blue. I really do want you to come back and always remember, most of the time our vacuole is going to store water. So our motor protein can take our chloroplast organelle and tuck it underneath that vacuole and hide it, help protect it. Well, let's say it's a cloudy day, or our plant is growing in the shade. What our motor proteins are going to do is they're going to bring that chloroplast up to the top of the cell and try and move it and arrange it and always keep putting it in the place where the chloroplast and ultimately the whole plant can get the best amount of sunlight. So this is like if you watch people on a sunny day who are sunbathing, and as the sun starts to track, as it starts to get hidden behind buildings, you'll see people start to move and follow that ray of sunshine. That's kind of what's happening inside of each individual plant cell. Or maybe you have cats. If you have ever owned cats and even some dogs, you will see that they always find the best ray of sunshine. And then as that sunshine moves across the carpet or moves across the floor in the room, they just keep scooching themselves over, trying to always stay in the sun. Our motor proteins with cytoplasmic streaming are doing the same thing with that chloroplast organelle. So our chloroplast organelle gets stuck in two categories. Not only does it do photosynthesis and make energy, but then it's also considered a plastid. And a plastid is going to be an organelle that's going to be storing stuff. So our chloroplast can store some glucose. It also has pigments, and a lot of our chloroplast organelles are going to have pigment molecules. So the generic name of a plastid will be a leucoplast, and the leucoplast can store a lot of things. It doesn't have to be real specific. So I'll draw a little leucoplast over here. Of course, when we studied starchy staples, and we looked at all of those starch granules underneath the microscope, what we were seeing is those starch granules inside of the ameloplast. So I drew that in black because remember, whenever we test with iodine, iodine is going to be interacting with the starches and turning it that deep blue, black color that we saw with our starchy staple lab. So our leucoplast could store anything. Our ameloplast is going to be storing starches. Now our other plastid that's really important this time of year, or any time of year when we have colored plants, is our chromoplast. Our chromoplast is going to be storing a lot of pigment molecules, mostly in those reds, yellows, and oranges. So what I have here today is I have this beautiful oak leaf. And what I love about these oak leaves is that here we can see there's still some chloroplasts, which is why we can see some green. But we're starting to see that green disappear, and what's showing us, or starting to show up now, is the chromoplasts. And those chromoplasts have, in this case, some yellows and oranges in the pigments. Now remember, whenever we see yellow and orange, we have to think vitamin A. Now what's happening in this plant is it's actually starting to take those chloroplast organelles and move them out of the leaf, move them down through the stem, eventually move them out into the trunk, and then store those chloroplasts in the trunk or even down in the roots. Chloroplast organelle is a really expensive organelle to make. And so by the time this leaf is done, and it does something called abscising, it leaves the rest of the plant. It's going to have that darker kind of reddish color. All that's left behind are the chromoplasts with all those pigment molecules. The chloroplasts are mostly gone and mostly being stored in the rest of the plant body. This is a beautiful thing about deciduous plants. They save some of the chloroplasts, but then next year, in the next year, they're going to have to, after they break dormancy after a winter or cold season, they're going to have to regrow all of these leaf structures. You're going to bring all those chloroplast organelles back out and start them photosynthesizing. It's really quite remarkable. A lot of plants are just going to get rid of their shoots above ground and then just rely on energy in the roots to regrow the whole plant. And then other plants, they just don't even try to make it through the cold season. They just die, they make their seeds. Good luck, kids. Hope that works for you. So these are some of the basic cell organelles. There's a few more that are covered in the chapter. Make sure you know what they're doing. We really in this class are going to focus on the nucleus, chloroplast, mitochondria, and then those plastids, like the ameloplast storing starches and the chromoplast storing some of those important pigment molecules, especially if they're indicating to us that there's vitamin A. Now in lab, one of the things you're going to do is you're going to be looking for a pigment molecule that's stored in the vacuole, and that pigment molecule is called anthocyanins. Now, anthocyanins are amazing, and anthocyanins are what's called oxygen scavengers. In other words, what they're doing is they're going around inside of the cell, and they're picking up loose electrons, and they're picking up any other free radicals that could cause some damage. Anthocyanins are part of the flavonoid family, and they could be helping to prevent damage to DNA. So in lab, you're going to be dealing with the purple cabbage. And that purple color tells us that not only do we have anthocyanins, but that those anthocyanins inside of the cells are going to be registering that the cells are a little bit alkaline, a little bit base on the pH scale. Some other amazing things you may not have thought about that also have anthocyanins will be things like the eggplant. So this eggplant, this deep, deep, deep purple color in the skin tells us that there's lots of anthocyanins in the skin. If you've ever seen a purple tomato, that purple tomato is related here to our eggplant. That purple in the skin has a lot of anthocyanins. Now the red tomatoes don't have the same level of anthocyanins. Purple potatoes, again, related to the eggplant, related to that nightshade family, having anthocyanins, giving it that purple color. Check out this purple carrot. These things are delicious and amazing. I don't know if you've ever had one, but look at how beautiful that is on the inside. Anthocyanins in this purple carrot. Now, interestingly, in a lot of our red apples, there's going to be anthocyanins here as well. And here, the anthocyanins giving us this red color tell us that the skin of our apple, at least, where those cells are located, are very, very acidic. Otherwise, we wouldn't get this deep red color. So anthocyanins are also a natural indicator of pH. Anthocyanins are neat. They're amazing. I hope you learn to start seeing them in the different things that you eat and consume, and even flowers. A lot of those deep purple flowers are due to anthocyanins. They'll keep coming up. They seem to be very important, not only for our own health, but also when we're looking at plants and their different qualities. And that brings us back to the cell. Because when we see an organ, in this case a stem, wrapped around an ovary, when we see an organ, we have to understand what is going on at the cellular level. So in this chapter, we focus in on the cells, how they're growing, the parts inside of them, how those parts are important, and then we're going to talk about the organs. That gets us to the plant body. It gets a little bit more complicated from here. As always, if you have any questions, please email me.